Huh, I could have sworn that that piece was going to come off.
Yeah, you would think so. It looks like it would.
I used to have a crowbar for some occasions,
Really
but
depends on how much you want that rock.
I don't know if the crowbar has actually been effectively used. Anyone commentary on that?
Yeah, I imagine how do you have leverage with a floating ROV?
Wedge it in somewhere, I guess.
Twist and a grab, and there it goes.
Can we get a tighter shot on that, Jeff, please?
Right? Rotate.
Yeah, thank you.
It's like a
It's
little
got some sponge
sponge
spicules.
bits. Yep.
Okay, yeah, it looks
Some
like what we've seen on other
oxidized patches,
parts of the seamount, right? That the top is a bit shinier and the bottom has this crumblier texture to it.
Right. Good capture. I think we can stow it.
Roger that. Full wide, please.
That's about, let's say, 10 centimeters.
All right.
And I believe it's dealer's choice on which bio box division you would like. And Jess will want to take a Niskin sample here before we pick up.
Yeah, you do. That sounds good.
Okay, for Niskin bottles. So again, Niskin bottles are our water collection devices.
So simple 80. We
have a row of open tubes on the side of Hercules here that are exposed to the water. Any free-flowing water moving through here goes through those tubes. But when the arm triggers one of the colored loops over here, it will, oh, thanks, Jeff. You can see in the lower quad, the top of those tubes, you'll see the top plug shut, and now any water in this region is captured in that container and will not mix throughout the rest of the dive. And that water contains any of the cells of organisms that are in this area. And what can those cells tell us, Beth?
Swing and a miss.
Well, our goal here is to see how the microbial communities in the water interact with the microbes that might be resident on these hard surfaces. So,
yeah.
yeah, we'll be looking at the diversity to figure out how much life is in the water, as well as the diversity of life in the water.
Yeah, so really able to filter that water, filter the DNA out of those cells and analyze it.
Ready?
Yeah, and that's one of the goals. Water will also be collected for a broader eDNA study, where the goal of that project is to understand how all these organisms in the corals and sponges, et cetera, that are releasing cellular material into the water, if they can be identified through water samples instead of through collection. And so this sample will also be investigated for that purpose.
Nice. Jess, ROV Jess, someone's wondering if you practice on the off season by collecting stuffed animals from claw machines.
Those are rigged, you know. That's potentially a very interesting training ground. But no, I do not, because that's a lot of quarters, and I still have to do laundry.
Quarters? These days they're like $1.50.
Are they $1.50 now?
But there's an iPad in there or something.
Rennie, can we go ahead and start getting ready for another ship's move?
Sure. It looks like perhaps still on long 135 we'll do.
Wow. That was a big
Bridge
heave.
now.
7 meter heave.
One step 50 meters, bearing 135.
Jake, were you just commenting that the heave is picking up?
Yeah, Argus moved about 7 meters.
Wow.
vertically
Really?
down.
Wow.
Yeah, so for our audience at home, the swell is starting to pick up out here. We are in big wave season in the region, so it's not surprising. And so we'll probably be ending this dive in just a few hours, and then having to head south to get out of the big swell.
We're still on for 4 p.m.
That's as far as I know. Yeah, we're still on for recovery at 4 p.m. Hawaii Standard Time.
Yeah, the way the weather's looking, it wouldn't be later. If it was earlier, it'd be because we're starting to see higher swell. Let's take a look at that weather.
This is beautiful in both views here.
The comment about the arm practice, there's actually a pretty cool toy, usually some sort of wood that you put together, this hydraulic arm, and they use syringes as the hydraulics, and so you can actuate each function via a syringe in and out. And it's really pretty close to what is happening here, just different controllers rather than the manuals. Syringes, it's actually very close to how the port side arm works.
Yeah, I think there's an activity like that in our education resources on nautiluslive.org. You can build, kind of DIY your own hydraulic arm. That's a good point. Wow, what are we looking at here on the broadcast?
That was a weather forecast.
Okay.
That was Rennie's.
Oh, you're on this.
Rennie's looking at. I got HIPAC plan and set.
Windy.
Yeah, we use a few websites. Windy is one, and Passage
Can we get
Weather.
a partial zoom on this?
Yeah, sure.
Ooh.
So there's a lot going on in that sponge.
There is a lot going on in that sponge. Jake, you want to give me a little scope there, please?
Yep.
So for our audience
Jack,
online,
do
if
a
you have any
partial there.
questions or comments for us, please feel free to use the chat box
That's
feature
great.
on nautiluslive.org.
We do have a question, wondering if we need any volunteers or interns. Do we take volunteers on the ship?
There are definitely intern opportunities with Ocean Exploration Trust. I'm not sure if they're open right now because there's a little bit of a backlog due to COVID. So we're still playing catch up with getting everybody out here. Oops, that's not what I wanted. But if you go to the nautiluslive.org website, and I believe if you click on the join menu tab, thanks Jess, that's great.
Pull wide, please.
There is information there for scientists, for educators, and for students about how you can get involved. And for the educators and students, there are fellowships and opportunities for internships. So definitely check those out. And as we heard on one of our late night 8 to 12 shifts, many of our current OET team started as interns. So just to give you a little encouragement of whether that pays off.
Yeah, you started as an intern, Rennie, and how many dives have you, or how many expeditions have you been on now?
This is my 55th Nautilus cruise.
Wow.
Dun, dun,
But who's counting, right?
Who's counting? My first one was NA134, and we're on 134, so I've done just about half of them.
Wow.
Very nice.
Which is a lot of days.
That's a lot.
That's a lot of days.
A lot of days at sea.
If you look at SAT-3, all of those people started as interns.
What was that there, Jeff?
Oh,
I said if you look at SAT-3, all of those people started as interns.
our whole front row.
Yeah.
Yep.
Plus Steve.
And Steve is waving.
And Steve.
Unfortunately,
the background.
Don't forget Steve.
Sorry,
the
Steve.
seat that Steve is in is almost as well hidden as my seat. It's really hard to get a picture back.
Yeah,
into
I
that
designed it that
corner,
way.
so.
Yeah.
No. Yeah, it's tough. But I think the best improvement in this van versus the old one is the homogeneity of temperature.
Oh, yeah.
Yeah.
We used to have very cold seats and very hot seats.
It seems to keep the computers cold and the people normal.
Colder?
I'm actually not that cold
Yeah, it
compared
used
to
to be
other vans
really-
that I sit in,
Yeah.
where I'm in my full main winter outfit.
Yeah.
So now if we look at SAT-3, we see that Steve, in fact, did not design a seat that cannot be seen from there.
I even have this awesome camera right in front of my face.
Jeff can go partial in on this guy here.
Yay. Hi, Beth. You
Yep.
can actually, in that view, see
Thank you.
our interaction in action.
Yep.
That's our studio where science communication fellows are able to interact with classrooms and learning groups around the world. We do free 30-minute sessions, talk a little bit about what's happening, and from that view, you can see the control van behind the window.
Thanks for those partial zooms, Jess
Yeah, they're fun.
and Jeff.
All right. Thank you, Jeff.
If you look now, you can see one of the interactions going on with the view of the control van.
So
Oh, thanks. It's
Okay.
all
almost to
connected.
this topographic high point, we've got another maybe 100 meters or so.
That is the view, Jeff, where most of the students that I talk to ask if that's a real background or not. It looks like a virtual background, but it is real. It's all
Sounds
connected.
good.
Assuming we get to the top of this high, where do we have to go next? Can we zoom on and head back to see what our
It's a
options
great
are?
call.
What was your question, Steve? Sorry.
No, I was wondering, assuming we get to this high point, where do we go next? What are our options?
Yeah, if you could zoom out a little bit, Rennie, on HIPAC.
We've got about 400 meters until our arbitrary end there.
Yeah.
That's kind of that, or choose one of these slopes.
And if you could zoom out just a little bit more
Yeah.
from there. Is that the extent of our multi-beam coverage?
Oh, we've got plenty, but that's the bit with the contours.
I see.
If
Okay.
you want to see the whole feature, I could keep going.
Yeah, right. Okay. I just wanted to check and see if we were in danger of getting towards a terrace or anything. So I think we could just keep going. But that probably will be the next shift to decide. And I think weather is going to be something we need to consider.
But this has been continuous from the start of the dive till now. This is pretty extensive high density and high diversity coral community, and
Right.
sponge community.
Yeah, a
So
lot of sponges.
A spiral pattern like that, is that just a different morphology of the same species, or is that a different coral altogether?
You talking
Yeah,
about
why
this
do
here?
you're talking about that one.
Yeah, that one versus the straighter ones.
Why do they do that? I think a lot of people ask that question.
Sometimes they just want to be different, Rennie.
Yeah,
You can do a partial there, please, Jeff?
you know, we asked this question about the Iridogorgia colonies a few days ago, right? Where we were
Right.
trying to figure out why they might do something like that. Look, you can see all the tiny squat lobsters on all these colonies now. They're everywhere. But these squat lobsters have their claws up into the flow, and they're just trying to capture anything that swims by. Sometimes it'll be a fish or something gelatinous. At least 2 squat lobsters there. Yeah, I think I wouldn't say it's an absolute diagnostic characteristic, but it's something that I use if I'm trying to differentiate certain types of coral colonies, bamboo coral colonies especially. For bamboos, you really need to focus on a
there,
couple of
please, Jeff.
characteristics, the sclerite structure and the polyp shape and size. The morphology is sometimes helpful, but bamboo corals do funny things. They branch when you don't think they're going to branch, and they just have very, very bizarre morphological characteristics on the colony. So what would be the advantage of something like that? I would guess, once you grow up enough off the seafloor, you can probably maximize your energetic efficiency in food capture by increasing the amount of time that your polyps have to capture food particles. So if you're larger, if you're coiled, and you use the friction of your colony to slow down the water that's moving past you, you have a better ability to capture small particles, for example.
Oh, it looks like a partially parasitized coral over there. And also in front of us.
Yeah.
That spirally coral was a bamboo coral, right?
Yeah, they're bamboo corals. Yeah.
Someone's had some nom-nomming on that coral.
Yeah.
That one
So this
pretty
hydroid
fouled.
Yeah, it looks like hydroids.
on it.
Yeah, but bamboo corals can do funny things too because of just something that's happened during their life history, for example. If they've experienced some sort of associate settlement event, like a anemone or something that may have settled on them, or perhaps, you know, bits of tissue loss that have been colonized by other animals. Sometimes the coral can fight back and recolonize area that it lost. And if, for example, if it creates a gall over something that has settled on it, like a, I don't know, name any animal that might settle on a bamboo, like a barnacle or something, and the barnacle dies, the coral can recalcify over the barnacle, creating a gall. And in some cases, that process might result in a kink in the colony, or it might result in, an inadvertent branch point. These types of things are what we as biologists are trying to understand when we're trying to determine what the abilities of coral colony are.
Would we be able to term this a coral garden, Steve? Is there enough?
Yeah, I think it fits the definition that the community uses for a coral garden: high density and high diversity. Yeah, I think you could apply that.
So it looks like we're kind of cresting a small hill here, which matches kind of with what we're seeing in high pack. There's a little feature, rounded feature on this northwestern spur of the Don Quixote Seamount. So it might explain the really high density here being at the top.
I'll echo what Lindsay said a couple watches ago. It's very Seussian.
Yes.
Or it looks like Chihuly glass.
Yeah, it does have that. You can definitely tell that some of the glass sculptures, chandeliers and things are inspired by nature.
The odd shapes and different forms that everything takes. It's cool.
Great comparison.
Definitely feeling some rolls up here in the control van.
Yeah.
Ship.
Yeah.
Some folks are, I think, saw our earlier watch. And just to clarify, this is not the same dive. We actually did recover those ROVs. This is a new dive on the same seamount, but a different location.
Correct.
And if you're just tuning in, we are looking at a number of different organisms. Can't identify all of them super quickly, but we're mostly looking at a community here of coral and sponges intermixed, and a beautiful formation coming up here. You can really see a high concentration, especially on the left side of this rock.
I think we've reached the
Yeah,
top
and
almost. Yeah.
Right now, Hercules has a almost southward orientation of the view. So that high density is kind of facing to the east, I believe. Trying to orient myself, which kind of makes sense with our theory about the prevailing current direction.
Sure.
Thank you.
Can we do a little like pirouette around the side of this rock?
Yeah.
So we can have the still camera get some images while it's snipping away.
Are we getting Argus shots as well here?
You want lasers on for this or lasers off?
I don't know. Can we do some beauty shots with lasers off?
Yeah, we can do it with the lasers off for sure.
Oh, the jelly and Argus.
Yeah, lasers off, please, Jake.
Lasers off.
Yes.
Thank you.
Yeah, these big primnoid colonies, I think these are it's a few
Give me a little more
species of
there, Jake.
primnoids here, but I think the larger ones right in front of us are Paracalyptrophora, but have only been able to get a Polyps open view of them, but that's my suspicion.
There's another one of those yellow features.
But there's also a number of unbranched bamboo corals. I've seen some black corals as well. Bathypathes is here. Zoanthids are quite common. Actually, you can see a couple different types of zoanthids. This mustard brown zoanthid, and then on the 9 o'clock position, we have these primnoid colonies covered with zoanthids. There's different species of zoanthid as well. Those look to be growing over in colonies of Norella.
Seems like a really popular spot.
Yeah, this reminds me exactly of a seamount that we observed.
Wow.
Oh, actually, you know what? I don't know if those are zoanthids. I think that's Stauropathes. Anyway, we can get a zoom after we're on these mustard brown colonies.
Yeah, definitely.
Right here.
Yeah.
I'm just gonna finish this pirouette because this is an amazingly large coral, and then I'll pop down.
Yeah.
Yeah, this is exactly the scene we saw at a seamount.
Looks good in the DSC too.
In the vicinity of Jarvis Island. We came up to the top and it was just blankets of this massive primnoid garden.
Mm-hmm. Oh,
Shrimp.
a little shrimp coming by to say hi.
Yeah.
Steal the spotlight, this shrimp.
5 seconds of fame.
Maybe come get a little snack.
There's no doubt that these colonies are quite old. Scott France at University of Louisiana
All right,
Lafayette is
Jeff, go ahead
saying
and push on in there.
the holdfast diameter is sometimes an indication of how old something is. So that looks like Stauropathes, maybe a black coral. I thought they were zoanthids from a distance, but looks like at least 2 black corals on this rock here.
Yeah.
There's zoanthid and primnoid colony just above.
And these little, right out here at 3 o'clock, that little yellow ring, that's probably an old holdfast and not some type of, 504.
Yeah. Yeah, they persist for quite a while.
Anything else you guys want to look at with the more zoomed up eye?
No, no more zooms.
No more
just
zoom.
kind of continue up and
I
around
wish
the
I
rock.
could say that for the whole year. No more
Sure
zooms.
No promises, but not in this frame.
Yeah.
Little crab up there.
Yeah, there's a cool
Squat
crab right
lobster
there.
yeah. Chirostylid.
Oh yeah, squat lobster. Look at that holdfast.
Yeah.
My goodness.
Wow.
Like a dinner plate sized.
Yeah, can we get the lasers on just for looking at that holdfast, please?
Yeah. Let's
Thank you.
pop down a
Yeah,
little
so
bit.
about 20, 25 centimeters.
If we were to put our lasers on it, yeah, let's see here.
Yeah. 25.
I think you're right, 25.
Yeah, right in there.
Very cool. All right, I'm gonna come forward. Start to.
But, I mean, in sonar, you can't see any other upward slopes, right? So this seems to be the top.
Yeah, I think this is the
it's
top
like
of
the
this
linear
feature.
ridge
We've
that we're on
summited.
top of.
There's another one over there.
Looks like we'll have a bit more.
It's all
of-
downhill from here, guys.
That is so beautiful.
I don't think that's true. I think we've still got more height ahead of us.
It's fantastic. Look at that colony.
We have a suggestion from a viewer for viewers to be using hashtag Nautilus Live or hashtag EVNautilus on Twitter, Instagram, Facebook. Thank you for plugging that. We would also suggest using hashtag inspired by Nautilus Live and hashtag Nautilus Live spelled out. Those are our aggregators. So thanks so much for following along. Thanks for taking your own screen captures and sharing this out. We would love to use you all, crowdsource this promotion of ocean exploration and inspiration.
That patch of white kind of looks interesting. Do you see that?
This in here?
Yeah.
Yeah, could
That's
we have
sediment.
a look at that real quick? Just
Oh, yeah, sure.
pivot over to the right for a moment.
It just looks like it's coating the rock.
Yeah, it looks like glue.
You know, in a way that's different than just sediment drape.
Yeah.
We're looking at the coral, right?
No, this sediment area
Oh, I understand.
has a weird texture.
Lumpy.
We're still seeing the telestrator out there, right? Yeah.
What's that?
You are.
Thanks.
All right, Jeff, go ahead and push on in there a bit.
Yeah, definitely some interesting texture to this sediment.
Down a little bit more.
Like clumps of spicules.
Yeah, it looks like just a lot of sponge debris has passed into this spot. So maybe the sediment is actually flat. It's just the sediment, the spicules give it a shadow effect.
Yeah.
Yeah.
Dunes.
All right.
It's kind of like an old, I would
I
imagine like an old rug too, that, you know, sponges, even when they're dead, they continue to accumulate sediment over time, just from stuff that settles on them.
All right, got to catch up a little bit.
Rennie, how are we doing on a ship's move?
Ship just stopped.
Okay.
I had called in another and gonna call in another. That's all right. Continue on.
So for our viewers at home, if you don't have the Grafana data feed up, we're at roughly 1,815 meters and continuing in a southeastward trajectory on this northwestern flank of the Don Quixote seamounts. And pretty much since this shift has started, maybe even since the dive has started, we've just had really incredible density of corals and sponges. As you can see here.
Got Grafana and SAT3.
Yeah.
Thanks. Thanks, Jeff. You can see a few of the stats that we're seeing: the temperature, average depth, Herc depth.
Oh yeah, thank you.
Lindsay, do you know if they have a link to Grafana yet on the website, or do they just? I know they have the depth and I think temperature that they display under the video, but do they have a link to that?
A link to what? Sorry?
The Grafana webpage?
I don't think it goes to Grafana directly. I think it's just an aggregator of some of the data. Like when you click on the more data.
I think this data is. I think this data is just local to us. It's not being sent out.
Okay, yeah, just curious.
Scientists ashore have access through a special portal to the Grafana data, but other than that.
Jeff, you want to do partial here?
Maybe that's what I was thinking of, yeah.
Yeah, it looks like some of these, you know, there's some grazing going on, like this bamboo coral to the right-hand side here.
Oh, yeah.
Looks like it's been grazed at one point, and then the animal gave up, maybe a sea star.
Got full or got too heavy.
Or it's regrown, potentially, the coral.
It's a large ear, this sponge.
Yeah.
If all the polyps
Right,
are stripped of a stalk, is there any way for it to continue growing?
You were very quiet.
Oh.
Maybe.
Steve, if all the polyps are stripped of a stalk, is there any way for that coral to continue growing?
Yeah, absolutely. It depends on if anything has colonized it since, though. So, you know, corals will just continue to produce polyps and overgrow hard substrate, especially if they're the ones that produced it. However, there are competing space resources in the deep sea. And obviously, if you're an organism that likes to feed in the plankton, you like to feed on suspension feeding strategies, you want to be high up off the bottom because that's where the water's moving faster and that's where you have a higher chance of capturing particles. And so a lot of other organisms like barnacles and hydroids, these types of fouling organisms will attach to their skeleton. We've seen that several times on this watch so far, and will attach and use that space, in which case it sets up a competition between the coral and the hydroid or barnacle or anemone, whatever manages to get there first. Zoanthid also. Zoanthids eventually, I don't know. Zoanthids are interesting because usually when a zoanthid starts to take over a coral colony, it becomes, it gets settled on a coral colony. It's a very slow process, but the zoanthid usually wins out from what we've seen. I mean,
Jeff,
yeah,
you want to do
I would like
partial on this guy,
I would like to be able to see how an invasion like that progresses over time. We have a suspicion that the zoanthid usually wins out because we see lots of these colonies covered with zoanthids only, but there's no way to say concretely that the coral doesn't have a mechanism of fighting back.
Now we're just getting a snapshot of the final product.
Very cool.
Yeah, thanks for also mentioning the hashtag Lu'uia Ahiki Ikapapaku and hashtag
please.
Papahanao Mokuakia Marine National Monument. Those are great hashtags as well. Again, we really appreciate those sharing of images on your social media feeds. Hashtag Nautilus Live, hashtag inspired by Nautilus Live. Jake, have you been using a slightly different strategy this dive with Argus with the changing and shifting surface conditions?
Not really. I mean, I guess I've maintained a slightly higher altitude or distance between Hercules. But yeah, we are seeing some pretty big heaves in Argus. It's come up to it's between like 4 and 6 meters, pretty consistently. You can see that in the Argus cam.
Yeah, you can.
Heaving up and down. It's more exaggerated than what the ship experiences because of the long cable.
Yeah, it's on a lever, so it's on the back end. So this is a moment where you can really appreciate the two-body system, and we've been, like, Jess has been just smoothly flying around, and we're rolling around up here, and Argus is eating a lot.
It's a very big hold fast.
Wow.
That's an old colony.
I was just going to say that, Rennie. Thank you. You can take the SCF position now.
Get an idea of the seas.
Do
Just
a
put
little partial here.
a mid cam.
Oh, thanks, Jeff. Yeah. If you want to see what we're experiencing.
Yeah.
Oh,
if you haven't already noticed our bodies rocking back and forth on the van.
This is a really nice keratoicidid bamboo coral. Take a look and see how it's branching. That might give us a better idea on
So dense.
Yeah.
what this belongs to. My suspicion is that, well...
Can we get a closer zoom on that?
Bring lasers off too.
Yeah, so this looks like a node branching colony.
Yeah.
And
you can see that here really well.
Primarily single branches at the nodes, but I did see about halfway up on the colony where there's that hole, there's a triple branch at the node. Or double branch, I guess.
And some of the branches look really thick and calcified. Others are much thinner.
That's an old, very healthy looking old colony.
Yeah.
Yeah.
Sometimes they have tissue loss at the base, but it looks like the tissue completely covers the entire skeleton there. J-clade, thanks Scott.
You can see the scale of it in Argus view.
This doesn't have as many associated organisms that are highly visible compared to some of the others that we've looked at.
Actually, let's pull right there.
Just have some kind of chemical defense to keep them off.
Or maybe it's, yeah. How do sponges reproduce, Steve? Are they spawners?
You know, it's been a while since invertebrate zoology.
Have we stumped the scientists?
So I'm going to have to defer to to Chris on that one.
All right.
Because I am not, I don't want to say incorrectly.
So hold that question for Hui Hua Kai. That is the 12 to 4 watch.
But maybe Scott can help me out because he teaches invertebrate zoology. He's in the chat right now.
Help us out, Scott.
Yeah.
So for the front row, just so you know,
Yep.
our expedition leader came in and mentioned that the plan is actually to extend the dive to 8 p.m. Hawaii.
Very interesting.
Captain agrees that what we have now is probably what we're going to have then. So our co-lead scientist suggests that we try to spend more time on this seamount, considering the high density. So just so our viewers know, it seems that our dive might be a little longer than originally planned.
Thanks for that update. That's exciting. This has been
Somebody
fun to see.
put more coins in the crane
I was
machine.
going to say, do we have enough batteries in
Yeah.
Hercules for this?
Which,
It's a reference to
as one of the scientists who has to be in the lab processing samples, I got to say I'm kind of excited to get a little more sleep for the next recovery.
There we go.
Yep.
Well, I don't know about the science chat, but the general chat is a buzz with coral reproductive strategies. So, or I mean, sorry, sponge reproductive strategies. Thank you all for coming through for us. Oh, we have some science chat info as well.
Yeah. You want to read it out?
Sure. Actually, do you want to summarize?
Sure. Let me take a look here.
Yeah, for our viewers at home that are not part of the Scientist Ashore team, just so you know, the scientists involved in this expedition are not just those of us who are on the ship, but we have experts all around the world who connect with us through a additional portal that allows them to see some more of the data and communicate to the scientists in real time.
Okay, so it sounds like it's a bit of a complex process for sponges. So they're broadcast spawners, both males and females. They release gametes and fertilize externally. But in some species, the females retain the eggs inside of the sponge, collect the sperm, but not consume it, and then transfer it to the eggs, fertilize the eggs, and then brood the larvae. And then when they're competent, presumably they're released.
Sorry, Steve, so there's some sponges that are brooders then technically as well?
Yeah, that's what it sounds like.
That blows my mind.
Yeah, it says they collect the gametes similar to food, just not digesting it, holding on to it.
Wow.
Wow.
Weird.
Yeah, I learned something today.
And when we talk about male and female sponge, so would an entire colony be one or the other?
Yeah,
so
I think that's the intention. The same is true for corals, in fact, too.
Okay. So this could be a she right here.
This one's quite curved inward versus
And
the other ones that are more
really
open.
quite large. So again, for our viewers at home, those green laser dots are 10 centimeters apart. So this sponge is probably a meter or so tall.
Yeah, I mean, you can see this Hercules as well, just the scale. Kind of tough view.
Let's see. You know what I forgot to do that I wish I had done was to drop a target at that pinnacle.
Oh,
Rennie, do you know where that is approximately?
I mean, it is kind of correlating. This entire lump here is kind of the pinnacle.
Okay.
We
Yeah,
haven't been seeing.
I don't want to guess then.
Yeah, it's pretty much this whole
The whole
peak is
contour.
is the flat top.
Because now we're clearly headed downslope.
A little bit, and then it looks like it'll pick up again with high pack.
Yeah, there's a little saddle.
Yeah, exactly.
We have a lot of excitement that the dive has been extended.
Great.
Sounds like some scuba divers out there have actually witnessed sponge spawning in shallower waters on night dives. It's interesting.
With the extended dive, unfortunately, we don't have extended capacity, so
Yeah,
we just have to pace ourselves.
exactly.
We
I was
have
thinking the same
one
thing.
forward bio box bin open and 2 compartments in the starboard box, 1 large, 1 small. The slurps are all open, and 2 Niskin bottles, one of which will correspond with the other rock collection that goes into A, and then one of them presumably before we recover.
So we're continuing to get some great insight from our scientists ashore on the topic of sponge spawning. And some of what we're hearing is that on this item that, the sponges are ingesting both food and sperm. And it seems that different species can recognize both their own sperm coming from their own species, as well as identify if it's coming from a different species, and then identify that as food. So some kind of innate immune response, and highlighting that, to our knowledge, deep sea corals sponge spawning has not been observed.
Can we
Jeff, you
zoom
want
in
to push
on
in on that
this?
sponge?
This?
Yellow thing.
Can we zoom in on this?
Right,
Oh, on the coral.
That, it might be something new we haven't seen yet.
Okay.
Roger.
Yeah, so sponge spawning hasn't been observed in the deep sea, but would probably look like maybe like a milky white
All right, go ahead
smoke
and push on
coming
in
out
there,
from
but
a
please,
sponge.
Jeff.
So we'll keep our eye out for that, in case we're lucky.
There we go. Something new. Maybe. We'll see. I have a suspicion we're going to want to get tight on this.
Trying to find focus all the way out there.
Let's try this. Sorry. We're set down here now.
Yeah, sit down so we're a little more stable.
Yeah. Stand by.
That ship move is complete, and I'll just wait on calling another one in. Argus has a little bit of movement left, but should
Roger.
still be able to stay here.
Actually, you want to come full wide there, Jeff? I'll creep a little closer.
That would
Oh,
help me.
while we're here, we got a nice view of maybe a crinoid attached
Be
to
able
a
to get a toe
sponge
in.
or a coral.
Yeah.
Wow, that background there is crazy.
Background's
Yeah.
nice, yeah.
Very Seussian.
Yeah.
It's a great,
Coral forest.
yeah,
I
a
have
wonderful
word on my bike.
screensaver view.
Okay, go ahead and push on in there, please.
All right, here we go. And can we turn off the lasers while we're zoomed in? Although I guess now they're not out of the frame anyway. Is that as close as we can get? Yeah,
I think this is a family we have not seen yet on this expedition entirely. So
really?
Yeah, this is my suspicion is this is a family called the Acanthogorgiidae. Of course, it could also be a plexaurid, but I'm leaning towards Acanthogorgid because it has a very dark axis and very sometimes yellow to white polyps. But it's the defining characteristics for this genus and family are often difficult to see while the polyps are extended.
Okay.
Yeah, so I think the consensus is Acanthogorgia, or which is really noteworthy. Neat little observation. Very small colony. They can get quite big, but what are we at now? 1,800 meters. So yeah, that's about the depth where we start to see.
Come forward, please.
These plexaurids and Acanthogorgids start to come out.
Sort of movement.
But yeah, Acanthogorgia, great.
And looks like we might need to move a little bit here to get back under Argus. Yep. So a nice observation. We'll keep our eyes peeled to see if we see that again. If we do see it again, would that be something to maybe prioritize sampling? Or is observation enough?
I don't know. You know, I'll throw that out to Scott and Meredith.
Okay.
They're on the chat here. And I'm not familiar with the Acanthogorgia collections in this monument.
Okay.
You know, I know we can identify to genus, but I'll have to double check on that.
Do you want to come up on the delta a little, please?
Yeah, I saw tether in, but
What's a spine crown on a
So
coral?
with this genus, the spine, they have these sclerites, right, that support polyp. They're embedded in the coenenchyme, the tissue of the coral. And in this particular genus, when the tentacles are retracted, it looks like spines coming out of the top of the polyp, kind of in a circular shape around the oral cavity, oral disc, around the tentacles.
So you'd have to get very zoomed in.
Yeah, you'd have to get very zoomed in. Yeah, pretty much right up on the thing with this camera. But we've had some pretty good instances of other vehicles with other cameras getting really nice shots of the sclerites when the polyps are retracted. In the laboratory, you can see it very clearly, yeah, with the eye.
all right, now that we're back out ahead, do we want to move along? Keep on going with our track?
They might be off their headset for a minute there.
Oh, okay, there is. Got plenty to look at, so we're fine.
Thanks. Yeah, if you're just tuning in here, we're on the Don Quixote seamount. This is our second dive on this particular seamount, but our most shallow and most northern facing dive on this seamount. We've just, again, received word that we will be extending this dive by 4 hours. So instead of recovering at 4 a.m. Hawaii time, we will be aiming to recover around
4 p.m. So now we'll be recovering around 8 p.m. Hawaii time.
Thank you, thank you.
Beth, would you like to continue on, or was
yes.
there? Okay.
Yeah, let's continue with our ship's move towards that arbitrary end target on HIPAC. Seems to have been rewarding so far. 135.
I really lost track of 12 hours there for a second. nowadays.
I think we're just so used to the late night shift that.
Yeah. Here in the control van, you know, we are not really able to see whether it's daylight or nighttime. There was a question about why we call it a control van when we're on a ship. I don't know the answer to that.
Yeah.
Shipping container.
I don't know that I'm the one to answer that question, why it's called a van. Any other front row or our camera crew guy has more experience with why things are called vans? Trucks?
These are just storage containers. That,
Yeah.
Very, very highly modified storage containers, but they arrived, two of them arrived on one flatbed, and then the third one, along with a chiller, another piece of equipment, arrived on another flatbed, but I don't know. It must be probably a European thing. Europeans use van to describe things like this, so that'd be my guess. I'm sure that somebody in the chat will say, no, it's because it stands for something.
I think also control storage container doesn't sound very good.
No, it doesn't sound really cool.
Doesn't roll off the tongue.
No.
I know that if we were going to make it more permanent, we were not going to continue to call it a van.
No.
Control room.
Control room.
Yeah. Jeff was instrumental in building it and outfitting the inside, not constructing the actual shell.
No.
And deciding where the cameras go.
Yeah.
That was actually kind of by committee.
By video committee.
Yeah, Dave, Ed, and I
Yeah.
decided, well, we'll put a camera here, and a camera here, and a camera here. And conveniently, none of those cameras can see the video guy.
Yep.
Like how it's one of the primary concerns, that making sure that the video person cannot be seen.
Yeah.
Hidden in the shadows.
Hidden in the shadows. I'm going to stay a little upslope just for the downslope here.
So we're getting some input from one of our scientists ashore that the yellow coral we were looking at a few moments ago, Acanthogorchid, has been observed around this depth range at some other seamounts within the broader monument area, but quite a bit further away. So that's great.
Looking at a photo now, it looks quite similar to one that was.
That's a good one.
No, it's just a photo from the musician seamounts.
Yeah.
But still, that's a good ways away.
Wow, we're getting some movement on Argus.
Yeah, I felt that.
Yeah.
Yeah, so we just, it looks like a fish is coming into view.
Oh,
Nice.
Some
nice.
Frankid
Good, and
cutthroat
push
eel.
on in on this puppy.
Little fish.
Right in the
close-ups
eyeball, so sorry about this.
are great. We're looking at dorsal fin characteristics. We're looking at lateral line characteristics. All these are important
And the
to
nostrils.
image document so that
Yeah.
our fish biologists ashore can take a look and get us some reasonable IDs.
Just because it looks eel-like and long and thin does not make it a true eel, but many of these common names may have
All
the
right,
word
full
eel in
light
it.
there, please.
Could be in the Ophiidid family, eel-like family.
So it's really interesting to me that even though we're kind of in a flatter terrain than where we've been in some places, it's still really high density animal life. So good food supply here.
Actually, for the flatness of it in the bathymetry, it actually has some pretty good slopey features still to it.
Yeah, that's true.
Some mounds.
Mounds.
Yeah.
Mounds.
Little rolling mounds. Gentle waves of rocks. Looks like we've got some, oh yeah, okay. Brittle star up there.
Yeah, and this all started as volcanic rock, Beth, but what, if had to guess how altered is the rock that we're looking at
There's
here.
a nice squat lobster and that Stauropathes in the background, the yellow mustard color at around 3 o'clock.
Over here.
Yeah, see that one?
Oh,
There's
yeah.
on this side of the colony, there's a really nice squat lobster. If we need something to look at while we're waiting.
Yeah, can we get a partial zoom of that?
What makes it a nice squat lobster, Steve? The
It's
size,
visible.
No, I just I could see it from this way away, so I suspect it's large enough for us to image from a distance. You can see red and white wash colors on it. And it's actually in a feeding posture, so tell us something about how squat lobsters feed. I wonder if this is one of the spiny ones. That's a different genus of chirostylid.
Oh, and a
photo bomb.
There we go.
I heard you thinking that was good to look at. Check me out.
Cuskeel.
Cuskeel, yeah.
Come a little wide there.
Those ventral spines.
Crash.
Well, hello.
I wish I could tell you some more information about the identification of that squat lobster, but most of the time I can get to family at best. I know that there's been some reshuffling of the taxonomy of chirostylids recently, so not quite up on the latest from that group.
Try looking at the slope the other way now.
All right, Steve, we have a kind of a repeat of the question and the discussion we had last night about sponge or coral skeletons. We're seeing some sponge skeleton here, but not a lot of coral skeletons. Can you speak to that?
Yeah. It's, so first, coral skeletons, they're typically much more fine. A big sponge is going to take a long time to degrade, but corals also, they kind of go through one of two fates. If the coral completely dies, the skeleton falls over and is, degraded over time. With bamboo corals, you start to see dissolution of the skeleton over time, where, the nodes, which are proteinaceous, they're apparent, but you can see that the calcium carbonate internodes start to shrivel and look like they're dissolving. And it's true. If there's no coral to maintain the skeleton, it will go back into solution, especially at these depths. There's some complex mineralogy associated with different corals and how how their skeletons dissolve, but for the most part, that's the ultimate fate. There's another fate similar to that, where sometimes coral skeletons can become encrusted with iron manganese crust, depending on the oceanography of the area, in which case sometimes they can persist for a little bit longer and be protected from that dissolution process. And then the third fate, I guess, is they just get occupied from another animal. We've seen a lot of zoanthids occupy skeletons, and they can keep and maintain and protect that skeleton from becoming degraded. But the one thing that persists, and we mentioned it earlier in the dive, is the holdfast. Even if the whole skeleton of the soft coral, in this case, is chopped off, you still have that heavily calcified disc that attaches them to the rock.
I don't know if we described the meaning behind our expedition name. Would now be an okay time for Kalamiu to weigh in on Lu'uaea